Hello everyone, a very warm greeting to one and all. My name is Sri Gauri and I would like to welcome you all for today's webinar on OmicsLogic Multi-Omics Research Program. So before we begin with today's webinar, for those of you who are joining with us for the first time, let me take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. From specialized training courses to research services that provide expert guidance and support, we are committed to providing comprehensive solutions for all your omic data needs. And so far, we have had the opportunity to work with coordinators from different parts of the world to offer bioinformatics training programs across 24,000 participants in 187 countries across different specialization tracks, such as oncology, infectious diseases, precision medicine, neuroscience, and so on. And in this webinar, we will introduce you to OmicsLogic Multi-Omics Research Program and learn how this program can help propel your research journey in omics data analysis. Now, this program offers an unparalleled opportunity to work independently on a research project using bioinformatics and data science. And alongside mentor support, you can delve into omics research, gaining hands-on experience to make meaningful contributions to the field of biomedical research. Now, if you're a student who lacks a prior background in bioinformatics, we also provide training plus research option, where students will first complete specialized training modules on our centralized learning platform called OmicsLogic Learn Portal. And this learning journey involves mentor-guided lectures, online courses, and example projects on topics ranging from introductory bioinformatics, genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python. And throughout the duration of the training period, we closely monitor and track the learning progress of students. And upon completion of the training phase, participants will be able to seamlessly transition into the research phase of the program. Now, during the research phase, participants will engage in a thorough literature review on their chosen research topic and work closely with the mentor to identify and access relevant data set for their research project. Now, for analyzing this chosen project data set by the student, we give multiple options. For instance, you can analyze the data without any coding at all. And for those interested in opting the non-code pathway, they will be given directions to access the T-BioInfo platform for bioinformatic processing and analysis of data. And this platform includes demo pipelines and analysis cloud infrastructure to run various bioinformatic pipelines. And for those interested in learning to analyze data through coding, we provide Google Colab notebooks, which will help you develop coding skills while learning popular packages in R programming and Python to visualize, annotate, and analyze complex patterns in biological data. And at the end of the project, students will submit their work to data repositories such as GitHub and publish their completed project on the OmicsLogic Learn Portal. Now, here's a quick glimpse into the various ways that students have leveraged these resources to work on different research specialization track. So, for instance, our Python training program was part of the Systems Medicine graduate program conducted at the Georgetown University. And the training ended with students submitting their research project on the OmicsLogic Learn Portal. And as you can see, students have developed machine learning models using Python to address research questions in various disciplines, including infectious diseases, metabolic disorders, neuroscience, oncology, and so on. And further, students also have the opportunity to participate in poster presentation competitions and win exciting cash prizes. And twice a year, we conduct Omics Research Symposium, wherein we invite experts from all around the world and provide their feedback for the students' research work. Now, there are also examples of student research publications, and here is one example of Prithvi, who was an undergraduate student, who conducted transcriptomic analysis on dataset from multiple sclerosis patients and published her research in an open access journal on immunogenetics. Simay Dolaner is another student who went on to complete an oncology-based research project on leukemia and published her research in Columbia Undergraduate Research Journal. And the student projects are not just limited to graduates and doctorates. For instance, Andre was a high school student when he participated in the research program, and he completed a neuroscience project that he presented at the Regeneron International Science and Engineering Fair, for which he won the Potemkin Prize. And these projects can also be showcased for higher studies and university applications, like some of our research students have done, such as Dilara Daikin, who got accepted to the Erasmus IMBRC program in marine biology, Dhruv Mehra, who got accepted to the Precision Medicine graduate program at University of Manchester, and Anusha, who got accepted to the PhD program. And faculties and researchers have also collaborated through our programs to publish their research work. And here's one of the latest collaboration that we conducted with a team of researchers at Neguchi Memorial Institute for Medical Research at the University of Ghana. And it is also possible to pursue an entrepreneurship and business innovation track, like a team of industrial researchers from a biotechnology company in Nigeria did. So they developed a microbiome test kit to improve the fertility of women in Nigeria. And such tracks are pursued not only by researchers, but by undergraduate and postgraduate students as well. For instance, this was a group of students who worked on developing an early detection kit for Alzheimer's disease using metagenomic datasets. And now we consistently share these updates on ongoing research project outcomes, student publications, exciting job opportunities, and latest article on our social media platform. We'll share the link to our LinkedIn group where you can explore all of these updates. So now I'll take you to our centralized learning platform, which is the OmicsLogic Learn portal that I covered in my previous slide, where you can explore the various courses and example projects that we cover. So here's the link to the portal. I'm sharing it in the chat box. So for those of you who have not created an account yet, just follow along the steps that I'm sharing right now. So here in the link, if you already have an existing account on the portal, you can simply enter your existing email ID and password and just directly log in. Or if you're someone who is creating an account for the first time, let me show you. If you're creating an account for the first time, click on the login option that you see here. And here, click on the create an account option. And then here you will be asked to enter a few details such as your name, email ID, password, your university, and so on. You can enter those details and create an account, or you can also log in using any of your existing social media handles. This could be your Google account, Facebook account, Apple account, GitHub account, or even Twitter. So since I already have an existing account on the portal, I'm going to click on login now and log in using my existing email ID and password. So I'll pause for a couple of seconds for the participants to log in or create an account if you have not created yet. And once you have created an account, please let me know in the chat box with a yes. And for those of you who already have created an account, please put a done in the chat box. All right, Jershom has already created an account, perfect. Uzair has also created, Sachi has also created. And along the way, if you're facing any technical issues in creating an account or you're not able to recover your old account, you can reach out to us as well, and we'll be able to assist you in creating the accounts. All right, we have multiple participants who have already created an account. Thank you so much. All right, so I'll quickly walk you through on the first steps as well as how you can explore this portal. So on the blue bar on the top, you will see welcome back with your email ID. So when you click on that, it will direct you to setting up your profile. So that is the very first thing that you need to do when you create an account. To be able to do that, just click on the profile button that you see here and make sure that your name is in proper capitalization. Since when you complete the courses and the programs, the name will be automatically generated on the certificate that you have provided in this section. And add a brief bio about yourself. So you could mention about your educational background and your research interests, and also link any of your social media handles. So in my case, I've linked my LinkedIn account and my org ID, and also add a profile image. So once everything is completed, it should be showing profile completion 100%. All right. And this is the activity bar where we track the learning progress of the participants and make sure that they are fit to move into the research phase of the program once they enroll. And under the courses tab, you'll be able to monitor the course progress that the students are making as well. And once the project has been published, that will appear under the projects tab here. And the programs tab will display any of the training programs or the research fellowship program that you are presently enrolled in. And once you complete all the required quizzes, assignments, and submission of the project, the certificates will appear under the certificates tab. So that was all that I wanted to cover in this discussion. So now to learn more about the research fellowship program structure, what are the various research specialization tracks, I would like to pass on the stage to my colleague, Ms. Sonalika Ray, to take over the session. Over to you, Sonalika.
Thank you so much, Srivari. Can you confirm if my screen is visible?
It is loading. Yes, now we can see.
All right, perfect. A very warm good evening to everyone present here, or good morning, good afternoon, in whichever part of the world you are. Hope you all are doing great. And I'll be today taking you through the research fellowship program structure. And we'll also discuss about the different possible omics analysis that you can work on, different research specialization tracks, also the flow of omics analysis that you would be working on, and different tools and techniques that you can use. And we'll also showcase you some of the data sets. It's not possible to go through an in-depth overview of each and every omics analysis in this session though, but I'll try my level best to just give you a taste of what all different omics fields and tracks you can work on. And if you join the program, what would the flow be like? So let's first of all try to understand what is the program all about. So as students embark on their professional journey with freshly completed degrees, they often wonder how the professional life will be different from studies. And many prepare for this moment in advance by exploring the possibilities to work in an area of interest through internships or research fellowships during their studies. And this process starts with a program that we have designed to introduce students or even researchers from all the backgrounds to the intersection of biology and data science, and we call it OmicsLogic. Not just students, even passed out students or PhD fellows or researchers who are from different fields, but they want to learn about a specific type of omics analysis are most welcome to join the Research Fellowship Training Program, work on a research project with one particular data set, get a taste of the analysis, get good hands-on with the analysis, and be all set to embark on a new journey. Now, what is the research program structure? So we follow the research program in 3 different steps. The first step is to get you trained. So you go through an introduction to different omics fields, you go through an introduction to different research specialization tracks if you're not aware of, and then you pick up one. We train you in that specific type of omics analysis, and then we get started with your research. So once you are trained in a specific field of omics analysis, after that, you review literature, come up with a research question that you would like to work upon. Or if you don't have a research question, we have a set of research questions and data sets that we have already prepared for you that will take you through towards the end of this session. You can be a part of that project, work on a project, and have your report ready. Now, this report that you work on could be for n number of reasons. One, it could be a dissertation that you want to submit in your college or university, or for experience, or for your PhD application or a job application, or if you want to switch your field from one to another, maybe get into bioinformatics with a specific type of omics experience that you want to get through this program. All right, so once you have your research question defined, you work on a research project closely with the mentor where you discuss your project flow, discuss your gaps where you are stuck while carrying out your research, discuss the methodology with your mentor. The mentor would guide you on each and every step of how you have to proceed with your research work, discuss what should be your next steps, and that is the way you proceed forward. And towards the end of the research work, you will have a report or a publication, whatsoever is your requirement. So that are the three simple steps we follow in the research fellowship program. All right. Now the question is, what all would you be going through? So when I said training or getting started, I meant taking you through an introduction to bioinformatics training program, and another course or program that we have put together is Bites and Molecules to give you a taste of different research specialization tracks. You pick a specific field. It could be genomics, it could be transcriptomics or single cell or metagenomics maybe, or multi-omics if you want to cover each and every aspect of omics based on your research question. And then you gain the expertise on specific tools and softwares that could be used for carrying out the analysis on your specific omics data type that you're interested to work upon. When I say tools or softwares, it could be computing languages, programming languages, R or Python, that you should be efficient with at least one language. If you want to succeed in the field of bioinformatics using or working with big data, and then specific tools and softwares associated with each and every different type of omics data that you would be working on. Now, what are the different research specialization tracks that you can pick? So once you have gained an expertise with a specific type of omics data, the next step is to pick up a field of research that you would like to work on. Either it could be oncology or precision medicine or infectious diseases or metabolic disorders or neuroscience or system biology or agriculture, space omics, or maybe cheminformatics. If you're confused which specific field to focus on, then you can definitely explore the OmicsLogic Learn portal where you have these different projects in different fields. You can explore them, get an idea. We are here to assist you with each and every different type of field, and then we can pick up a question for you. All right. So that was all about what the structure would be like, what all different research specialization tracks you can pick from. Now, what's included in the program? A question that a lot of you ask. So you start with the training process. In the training process, you will be given recorded lectures. You will be given asynchronous coursework that you can go through at your own pace. We understand that you might be working professionals or students and might not have a specific time that you can dedicate to the live lectures. That's why we have given you recorded lectures and also asynchronous coursework. Go through them and then get back to us with any of the doubts that you might have. You'll be supported with the T-BioInfo platform to learn how different pipelines could be used for carrying out omics analysis. I'll also tell you what are the capabilities of the platform and how you can leverage it the most. And then you will be given R and Python code collabs as well to get a good hands-on practice with R or Python collabs or code blocks for different types of analysis. Guidance by these guidance by mentors, experts on specific research projects that you'll be working with. And towards the end, when you have to work on a research publication, let's say, or simply have your dissertation put together, we'll be assisting you in writing your manuscript or your dissertation project, and you could get a recommendation as well for your applications that you would like to push for your master's or maybe PhD. And you also can take assistance for any proposal writing for the grants or maybe research projects that you would like to pursue. Now, there are different research applications of omics studies that we discussed, and different omics studies as well that I just mentioned, that you can either pick one of these omics studies that you would like to work upon. So I'll take each one of these omics types one by one, and let's just try to understand what type of analysis you could do under each one of these omics fields. So the first one is genomic data analysis. For genomic data analysis, you could have these FASTA or FASTQ files. From different repositories, you take these different files and perform two very specific type of analysis, either phylogenetic analysis or variant calling. There are more such research questions that you can cater to with the genomic data that you get from public repositories. This is just an idea that you can start with. Now, there are two things that I would like to discuss. One is the code platform or the code pathway, and another is the non-code pathway. So for the non-coders, core biologists, we have these pipelines put together that you can run through and get your results and focus more on biological interpretation and reporting your findings rather than the code analysis or the coding that you might not be well equipped with. And for core coders or bioinformaticians who want to follow the code path, we'll take you through the training of R or Python if you're not well equipped with, and then we follow the code path to answer your research questions. Okay. Next is transcriptomic data analysis. I'll be explaining the non-code path and code path for each one of these omics fields. For transcriptomic analysis, you'll be taught in your training how to carry out RNA-seq analysis right from having the data in hand to how to prepare the data, how to explore the data. Then you go to the downstream analysis and then towards functional characterization or further biological interpretation. If you follow the non-code path, you have the T-BioInfo platform where you would like to know what type of pipelines you could run. When you talk about the pipelines, the first question is what tools am I going to use? And for those tools, what those tools do? Why would I be using a specific type of tool and not the other? And also, what parameters do I use? On what basis do I set those parameters? So all these questions would be answered during the training process where you will be taught about each and every tool, how these tools vary, why do we have, first of all, different tools for one single type of analysis or maybe step in our analysis. And then very usual Excel, how to use Excel for statistical analysis. One very common thing that we have seen in biologists, they are not able to use Excel for general statistical analysis. And also, how do you use or go through the code pathway? How do you use R or Python for carrying out the analysis, for exploring data, for carrying out the analysis? And if it is transcriptomic analysis, then how do you carry out biological interpretation, downstream analysis, and functional characterization using R or Python? Now the question is, where do you find such data? Because if you are students or researchers who are interested to learn about a specific type of omics field, but don't have such type of data, where do you get it from? So there are definitely a lot of repositories that you can explore. The question here is, how do you extract the data from these repositories? And most importantly, how do you find the right data set that you can use for your research project? Now, there are many data sets for which already a good research has been worked upon. So why do you even pick that data set? What type of research question can you answer with that data set? And how do you extract this data? Make sure that the data is accurate for your analysis, prepare that data, and then there are several other steps that you need to follow. Explore the data, try to understand what samples do you have or what is the variance between them. I'll just go through it step by step. So these databases could be GEO, Gene Expression Omnibus, which stores the gene expression and molecular abundance data, or TCGA, which stores the genomic and clinical data. You have FireBrowse that will provide access to TCGA data analysis tools. SRA is there, that is Sequence Read Archive, that will give you the access to raw sequencing data from various high throughput sequencing platforms. Expression Atlas hosts gene expression data from different species and cell types, and ENS stores nucleotide sequence data, including raw sequencing reads and assembled genomes as well. And then you have GPR, that is Gene Expression Profiling Interactive Analysis, which stores or offers gene expression analysis based on TCGA and GTEx. And then you have the Human Protein Atlas that contains the protein expression profiles in human tissues and cells. And these are just some of the databases that we'll take you through. There are more such databases that we'll cover in the training program for pathway analysis or gene ontology or functional analysis maybe. Now, as I said that you have these FASTQ files or FASTA files. Once you have these FASTA files or FASTQ files and you want to generate a gene expression table, if you don't have the facility of a good high computing system that you could use, or if you don't have a proficiency in Linux maybe, or a bash where you can generate a gene expression table, then we have the pipeline on the T-BioInfo server that can generate the gene expression table for you from the FASTA file or the FASTQ file. Now, what do I mean by generating a gene expression table from the raw read? So this specific this specific step is called pre-processing of reads into a gene expression table, and it is crucial for removing the noise or the biases and the artifacts, which ensures accurate downstream analysis. Now, there are several challenges that include data heterogeneity and quality as well, which the T-BioInfo server pipeline addresses through standardized pre-processing protocols and quality control measures as well, which enables reliable and reproducible analysis. Now, there are two set of students or researchers that we have observed. One who want to follow the non-code pathway and the other ones that want to follow the code pathway. The non-code pathway, this is the apt pipeline that they could use. And for the coders, we soon have a program launched in Bash where they could learn how they can generate a gene expression table. Now, coming on to the next step, that is data preparation. Once you have the gene expression table in your hand, what next? The next step is data preparation. Data preparation of this gene expression table involves processing raw sequencing data into a table format that will list the genes and the corresponding expression level. And this process will include the steps such as alignment to reference genome quantification expression level. And then you have to normalize your data to correct for biases, filter it to remove low quality or irrelevant data. And then you have the final gene expression table that serves as the basis for downstream analysis. Before even getting into the downstream analysis, it's important for you to explore your data and understand what types of samples do you have, what are their characteristics, what is the variance between the samples if there is any, and is it good enough to proceed ahead for downstream analysis or not. So exploratory analysis, basically it's an approach to analyze the data sets to summarize their main characteristics, often employing different visual methods. It involves techniques such as plotting histograms or scatter plots, box plots to understand the distribution, the correlation, and patterns within the data. Exploratory analysis basically aims to uncover these insights, identify the patterns, and also detect the outliers that provides a foundation for further analysis or hypothesis generation. After exploratory analysis, you can perform dimensionality reduction to look for the specific outliers and understand the sample variance. And from simple bar plots to complex dimensionality reduction techniques like PCR clustering for data mining, you can also use several clustering algorithms to cluster the samples together and try to understand if there is any batch effect maybe in your study, and if it is well enough, accurate enough to proceed ahead for downstream analysis, or if you have to remove some of the outliers that you might have in your study. So this is what the server pipeline and the results look like. And on the server, you have these files that you would get to download. So on the server, you will also have a dashboard, and it helps you interact with your results and change the parameters real time and see how your results vary or change so that you could report the findings. After differential expression, the next step is functional characterization, where you want to understand the gene ontology, understand the affected pathways or the genes as well. So you perform pathway analysis and functional annotation too, and also look for the specific type of differentially expressed genes in the pathways as present. So that was about transcriptomic analysis. an overview where you could use the automated pipelines or an alternate method where you could learn how to go through R and try to replicate the entire process using code blocks yourself. Have your file generated and have your results generated. I'll take you through a collab to give you a taste of how transcriptomic analysis in collab looks like. Next field is metagenomic analysis. For some of you, this might be a new field, but a lot of research is being done in this field these days. So metagenomic analysis is a method that is used to study the genetic material that is recovered directly from the environmental samples. And this approach is particularly useful for understanding the microbial communities, which includes bacteria or viruses and even fungi, in many of which that cannot be cultured in the lab. And for metagenomic analysis, there are two major pipelines that are being used. One is the data2 pipeline, and another one is the KIME2 pipeline. And for these two pipelines, you get to know what are the microbial abundances in your sample, what is the abundance of different microbial communities in your sample or between your samples, and also you get to know about the alpha diversity and the beta diversity within the samples. This is just an overview of what the field metagenomic is all about and what type of analysis could be done. Also, phylogenetic analysis could be done on metagenomic studies that we can talk about more in detail sometime later. So that was about metagenomic analysis. One way is you use these automated pipelines, and also we have proper coursework put together using R for metagenomic analysis. So what are the specific packages that are to be used, and what type of plots you can create, what type of analysis you can do using these specific packages is what we'll take you through in the training process, and then you work on your project. Now, another interesting field is single-cell transcriptomic studies. Now, here I want to give you an example of what single cell are to make you understand what single cell RNA-seq study is all about. So just think of bulk RNA-seq as extracting the juice from a mix of fruits, where you get a delicious blend but lose the individual flavors of each fruit. So it's like getting a broad view of gene expression in a tissue where everything is combined. And now imagine single cell transcriptomics as examining each fruit separately, savoring the unique taste and also the character of each one. So it basically allows you to zoom in and understand the gene expression at the level of individual cells, revealing their distinct identities and behaviors within a tissue. That is what single cell transcriptomic studies is all about. Here you have the single cell data, that is a set of computational techniques used to analyze and also interpret the data that is obtained from single cell sequencing experiments. And unlike traditional sequencing methods that provide us with an averaged signal from many cells, single cell sequencing will capture the genetic information from these individual cells. And the individual cells, as I'm showing you an example here, that it allows you to understand a more detailed examination of the cellular differences and also gives you a better understanding of the complex biological systems in terms of what is happening inside each and every cell. So this is what the single cell analysis pipeline looks like. It's quite complicated. Single cell process is quite complicated. We have tried to simplify this for you. If you are one of those students or researchers who is interested in the single cell studies, we'll take it through this process where we'll first of all try to help you understand the entire process, what the workflow of single cell studies look like, and then also we'll dive into the R and Python scripts to develop these different plots to understand the heterogeneity of cells. Single cell data analysis is a rapidly growing field, and due to its potential to look for the cellular heterogeneities in healthy and diseased tissues, it contributes to advancements in precision medicine and our overall understanding of biology at the most fundamental unit of life, that is the cell. And As students, you can also conduct research projects around different topics on cheminformatics or computer-aided drug designing as well, and address questions around the integration of different biological and chemical data sets to discover new drug targets maybe, or look for drug molecules that could be repurposed or perturbations, or also use of molecular modeling to simulate and optimize the drug protein interactions, also perform virtual screening for rapid identification of potential therapeutic compounds, or maybe perform network analysis to elucidate the complex biochemical pathways, and the application of machine learning to predict the molecular behavior maybe based on chemical structure. So this is also one of those areas that you can work on. And these were all the different types of omics fields that you can pick when you want to work on your research project. Now comes the tools and the techniques that you can use. So for all of these different omics fields, there are several uh, tools and techniques that you can use to analyze your data. And I want to talk here about data science and machine learning, as data science has significantly enhanced biomedical data analysis research by enabling the extraction of meaningful insights from the large and complex data sets. It facilitates the identification of disease biomarkers. It helps you predict the treatment outcomes or maybe personalization of medicine through advanced techniques such as machine learning or statistical modeling and bioinformatics. So we'll take you through a process where where you learn about what are the different types of machine learning, what are the different types of algorithms used under each type of these ML methods, be it supervised, unsupervised, or reinforcement learning, and what are the different types of tools that you can leverage under the field or the scope of data science to extract meaningful insights from your omics data. Now, I want to address here one of the very main questions, that is why T-BioInfo platform. So I want to explain here the difference between how using the T-BioInfo platform can be helpful and how using the code pathway could be helpful. So the T-BioInfo platform is designed to help researchers work with big omics data more efficiently. And it does this through pre-built pipelines that automate the complex analysis tasks, such as genomics, transcriptomics, and more. And these pipelines save time and effort, allowing researchers to focus on the biological interpretation of their results rather than getting bogged down in technical details. And the T-BioInfo platform is especially useful for non-coder biologists who may not be familiar with running complex code or setting up high-performance computing systems, maybe. And instead of spending time learning the code languages or managing computational resources, researchers can use the T-BioInfo's user-friendly interface to perform advanced analysis that would otherwise require high computational systems. So this makes T-BioInfo an ideal tool for students who are working on projects with tight deadlines, as it can help overcome the technical barriers and also complete their research in a shorter time period. And for the coders, we have the pathway defined. Take the data, learn how to use specific programming languages that you are interested in. It could be R or it could be Python. Also, based on your research question, what it demands, what packages in R or Python could be helpful for your specific research question. And then you have your analysis performed. So these are the two major pathways that we'll take you through in your research fellowship program. Now, I would like to take you through some of the data sets that we have put together. Those are literally just few of them, just to give you a taste of what type of data sets can you work with. Before I get into the data sets, this is what I wanted to show you. So when I say that we also follow the code path, how do we follow that? We usually work with Google Colabs because working with Google Colabs is quite collaborative, I'll say. If you're working with a specific code block and if you have some specific question or you encounter any error, we can simply exchange the links and we can assist you accordingly. So this is a Google Colab for transcriptomic data analysis. I took you through a brief overview of what the workflow for RNA-seq analysis looks like, where you look for differentially expressed genes, you generate a volcano plot, you look at the up or the down regulated genes, and then you carry out pathway analysis, and then you look for the affected pathways or the genes as well. The plots are not showing up right now because it have to be run, but you can create different types of plots here to understand what type of pathways your DEGs are involved in, or also learn about the gene ontology of your DEGs. Okay, so that is how you can work with R or Python. Now, how do you learn this? This collab can be specifically used while you're working on a research project, but how do you start this learning process? So as Shri Gauri already introduced you to the OmicsLogic Learn portal, we have put together here different courses on different omics fields and different tools and techniques that you can use to analyze your big data. Now, to get started, if someone is interested in learning R, so the first process would be to take you through the R coursework. We'll not just give you this collab to start working on. We'll definitely train you first of all to so that you have a good hands-on with R. You are well familiar with what packages to use, how to use, what should be the scripts, etc., etc. So I'm just giving you an example of one of the lessons here, what the lessons inside the coursework looks like. So inside the lessons, you will have the explanation of the package. Let me zoom my screen a little bit. Inside the lesson, you will have an explanation of what packages are being used, what do they do. Then you have the syntax given, an explanation of that specific syntax. And then for different visualizations, what the code should be. And then you have these interactive plots given here that you would get from the code blocks. Now, it's also a challenge for some of the biologists, I'll say the beginners, the newbies, to be able to understand or interpret these plots. How do you interpret the results from these plots? Just getting the plots is not like what we want. We want to understand the insights from it as well. So you have the explanation of the plots given as well, of what these plots are trying to say, or what type, what interpretation can you make from these plots, right? So that is what is being explained. You have here different visualizations. I'll just give you a quick overview. Different plots. You complete the specific step, histogram, density plot, heat maps. Towards the end, you have some practice codes as well. And once you have a good hands-on with a specific type of code block, and only then you are taken up next to work on your research project using the same code block. So that was one thing. Another thing is, there are several open source tools and databases that you can use as well. Inside the databases as well, there are several tools that you can use for analysis. So we'll also take you through the process of going through these different databases, tools integrated inside the databases, and different tools, open source tools that you can use that could be helpful to you for your analysis. Now, let me give you a glimpse of some of the data sets. So we have shortlisted some data sets from different omics fields for genomics, metagenomics, transcriptomics, single cell, and also the chem informatics field. We have some data sets and structures ready that you can take up and start working on for your research project. As January is that time period when many of you have to work on your dissertation projects, you can leverage these data sets or work on with a specific analysis with us. Now here you have two options. Either you want to work on this specific research project independently, or if you are a group of students and you want to work on a group of your own, or maybe if you want to work on a group, then we can have your group here. So a group project or an independent project, both of the possibilities are there. Now let's talk about those data sets that we have shortlisted that you can work on. I'll not go in much depth of it. I'll just tell you what topic is for the data set. And if you have a specific interest, then you can reach out to us, or it might also happen that I might not discuss a specific topic of your interest. Let us know if you have a topic of your interest. Maybe we can look for a data set for the same and then help you frame your research question and work on it accordingly. So the first data set is on cervical cancer samples of patients, and it has got 48 samples. And the another one is this data set is one of my favorite data sets that we have been working on to explore the different uh, multi-class samples. It has different blood platelets samples from lung, pancreas, breast, and also HC or GBM samples are there, CRC samples are there. So it has got good number of samples, and we could together explore these different samples and try to extract insights from it. Another data set that we can work together is on SLE, and it's an autoimmune disorder. And then you have another data set on metastatic and localized human pancreatic neuroendocrine tumors. This is one of those data sets that we have the research questions ready already for it. So the data sets that I'm showing you right now, we have the research questions framed for these data sets, obviously different from what was addressed by the researchers in this paper, but we can definitely leverage these data sets as a lot of more insights could be taken out from the data sets that are there on the public repositories. And then another one is on gastric cancer, diffuse type gastric cancer to be more specific, and lung cancer, where you have the lung cancer and control samples. And the sample size is 91. Now, apart from GEO, we have also taken out some samples, shortlisted some data sets from ENA and SRA as well. So on ENA, there is a very beautiful study on metagenomic data, which has 1,000 subjects. It's an Indian data set, and it has got different factors such as BMI, lifestyle, dietary habits, and genders. And this can be a very extensive and nice study, taking into consideration these different factors and explore the microbial abundances um for the all of these different factors that we could analyze. And we have more research questions based on just one data set that we can address. We can discuss that based on your interest. So if you're interested in any one of these data sets, you can let us know. We can discuss the research questions that you can pick up. And the last one that I want to take you through is the SRA repository. On SRA, there are several metagenomic and genomic data sets that you can explore. As time is a constraint here in this webinar right now, that's why I could not go through a lot of data sets, but just giving you some examples of what type of data sets can we work upon. And here is another data set. It is again from Indian population to study anemia and the gut microbiome composition in rural Odisha population. So these were some of the data sets that I wanted to show you. And if you have a specific interest, a specific topic of your interest, feel free to reach out to us. You can also explore these repositories and look for a data set maybe, or if you have any issues exploring the repositories, you can get back to us and we can have your research question framed. That was all that I wanted to discuss. If there are any questions, please feel free to post it in the chat box. And then I'll be heading it over to Shubhang.
Thank you, Sonalika. So, hello everyone. I'm Shubhangna from OmicsLogic. So here I'll be.
Shubhangna, your voice is feeble. Could you be louder, please?
Am I audible now, Sonalika?
Yes, better. Thank you.
Okay. Hello everyone. I'm Shubhangna from OmicsLogic. So at OmicsLogic, we believe in offering an extensive and flexible research journey for you. So I would like to give you a brief overview about the research program that we offer. So here it is. So the research program we have is in 3 levels, that is beginner, intermediate, and advanced one. First of all, I'll start with the beginner one. So beginner works on predefined research topics, gaining experience in research projects, designs, and omics data analysis. So you will gain hands-on experience in project design, omics data analysis, and we will take you on a journey of data collection, mastering data validation, interpretation, and project presentation as well. It is a perfect kickstart for your bioinformatics journey. So you can get started with it, and the access will be provided for 2 months. Next, we have is intermediate level, which focuses on exploring various research opportunities for you using various omics data types across specialized fields. Building upon the beginner's foundation, this intermediate level introduces research specialization tracks in infectious disease, neurosciences, metabolic disorders, as Sonalika told you already. So next we have is advanced level. For those who are seeking a deeper understanding, advanced level focuses on publication ready research, covering advanced tools, data science, R, machine learning, and programming in Python as well. So we help you in your publication journey as well by reviewing your research manuscripts. So OmicsLogic, in short, provides a dynamic, customizable journey for you through the world of bioinformatics. So now moving on to the pricing of the program, we have different pricing. I'll show you by, first of all, I'll be showing you in USD pricing. So for the research duration of 2 months, there you can dedicate 2-3 hours a day. The cost for research infrastructure will be $350, and for the students who are applying in groups can be enrolled for $300, and for various scholarship opportunities you can contact us at communication@omicslogic.com. We will definitely provide you with several discount opportunities and research scholarship opportunities available. So for 3 months duration, you can enroll for $700, and a discount scholarship for students participating in groups will be $550. And for 6 months duration, it will be $1,050. And for the students participating in groups can be enrolled for $900. Now I'll be showing you the pricing in INR. So pricing in INR will be 24,000 for a short project, and scholarship for university or groups will be 20,000. Similarly, for the 90 days project, it will be 33,000 and 28,000 INR for group projects. For the advanced level research program, it will be 42,000 INR for 128 days, and it discount can be provided like 35,000 for the group projects. And for 180 days, if someone wants to go for publication, they can enroll for 60,000, and the discounts provided will be 45,000. So similarly, here I will show you on your program page, you can change your country from here. And you can check your pricing in your region. For example, I'm changing it in Nigerian currency. So you can see the pricing from here in the changed currency. Similarly, in that way, you can check the pricing for INR, USD. So now I will tell you about frequently asked questions that are asked by students. So for scholarships and other discounts, you can contact me at communication@omicslogic.com. So I'll provide you a customizable plan that suits better for you. So what is the frequently asked questions that are asked by students with us? Firstly is, who is the program designed from? So the program is designed for students in high school, graduation, post-graduation, working professionals in the fields of bioinformatics, biotech, pharma, nutrition, health and wellness, and diagnostic industries. So next question that I hear from students is, can I utilize this training as requirement for a university internship, major project, minor project, company internship, or industrial training? So yes, definitely you can use this research program for it. So we have different projects that are completed by our students, so you can review it on our website. So I'll paste the link in chat box. So from here, you can review various projects that are completed by our students on in silico analysis of affected biological pathways, transcriptomic analysis, and many more. So the next question I get from the students is, what is the difference between 2 months, 3 months, and 6 month research program? So the 2 month research program is tailored for beginners who want to start their career in bioinformatics, research with predefined data sets and questions. Those students can enroll for 2 months per program. And for the students who want to opt for 3 months, you will initiate a research proposal and you can choose your own data set. And you can also work on the predefined data set as well. And the 6 months program is tailored for the participants who wanted to conduct advanced multi-omics research. So that is a perfect way to start your bioinformatics journey. So next we have is, after completing my research program, can I publish my research work or submit it as a dissertation to fulfill my academic requirements? Yes, absolutely. You can submit your research project for publication. And we also provide you with help with publication by reviewing your manuscripts and provide you a better journey for publishing your research. So that's all from my end. So if you have any questions or queries in mind, you can definitely leave them in chat box and I will review your queries and tell you the details for it. Thank you.
Yes, Shubhanga, we do have a few queries in the chat box related to the program. There is one query about scholarship opportunities for the participants, and especially for the international.
Okay. So for the international students, you can email me at communication@omicslogic.com. So we will offer you a tailored, tailored project detail. So you can enroll. I'm pasting my ID in the chat box. So you can reach out to me with your research interest and I will provide you with the appropriate information. Furthermore, we can also arrange an exploratory call for you. I have pasted my email ID in the chat box and you can reach out to me. Thank you.
Thank you, Shubhanga. If anyone else has any other queries related to the program structure or duration, please feel free to either type in the chat or you can unmute yourself and pick up your query. We have shared the email ID in the chat box.
Thank you so much.
Yes, go ahead.
And I wanted to update one thing, like we have installment options also. So we can provide you with tailored installment options according to your budget.
There's a question by Kurai. Kurai, we don't have a Slack or Discord, but we do have a WhatsApp group and a Skype group. We'll share the links. You can join them.
I'll be pasting WhatsApp community links in the chat box, so you all can join our WhatsApp community as well for further updates.
Right. Yes, there was one more query related to the short multi-omics workshop that we held last time. So that was a limited time workshop that we did, and that actually was really, but we will be having
Thank you very
more
much.
such workshops in the upcoming months. So you can stay tuned and follow us on our social media channels. And if there are any updates, you will be able to definitely inform. I hope that answers your query.
Thank you.
Sorry, Sitli, can you perhaps type in chat? I guess there is some background equipment. All right, and if anyone is interested in having one-on-one exploratory calls to learn more about the research program and explore your research project, you can simply email us, and then we will conveniently schedule a call on Zoom or Google Meet to address your query. We'll share the email ID once again, and all right, if anyone has any other queries, please feel free to reach out. And if no more questions are left, we'll end the meeting. All right, then. Thank you so much, everyone. And we look forward to seeing you on the research program. Have a great day. Bye.
Thank you for joining, everyone. Bye.